Using protein databases to find suitable positive and negative controls. Nowadays, there are several useful protein databases that can be used to find suitable positive and negative controls for your experiment. Here, I will go through a few of the commonly used databases such as UniProt, GeneCards, the Human Protein Atlas, Untitled, and the Cancer Cell Line Encyclopedia, each which have different strengths. As a first port of call, when researching a protein, UniProt can prove very useful as it has a lot of information regarding the protein function, subcellular location, and the different isoforms, expected molecular weights, and any modifications which may be useful to know, especially when analysing using Western blot. On the screen, you can see the database layout where you type in your gene of interest. Here, I am looking at the gene CXCR4, and it gives me information on the protein function. By scrolling down the page, there is plenty of detailed information regarding the protein. Here, I have chosen to look at the subcellular location of this protein to see where it should be expressed in cells. GeneCards is complementary to UniProt and also has information about the subcellular location. To find the subcellular location on GeneCards, scroll down the page to the localization section, which shows a cell coloured in different shades of green. The darker the green, the higher the protein expression. Here, for CXCR4, you can see a high protein expression in the plasma membrane, the extracellular space, and the lysosomes. Subcellular location can be very useful in determining whether an antibody is specific or not. In this example of CXCR4, you can see that the protein should be predominantly expressed in the plasma membrane. In immunocytochemistry, this is quite easy to see as all of the staining is on the plasma membrane and is minimal in the nucleus in Jurkat cells. In Western blot, it can sometimes be useful to run different cell fractions to check that the protein is expressed in the right cell compartment. Again, here we can observe a band in the whole cell lysates, which is concentrated in the cell membrane fraction and is absent in the nuclear fraction, as is expected. GeneCards can also be used to look at mRNA and protein expressions in different cells and tissues. This information is extremely useful when deciding what positive and negative controls to choose. If possible, you should test the antibody on a panel of lysates, choosing some lysates with an expected high protein expression, which will serve as your positive controls, and others with a low protein expression, which will serve as your negative controls. I will illustrate this with a second example. If I were interested in a different protein, for example, in the protein c-Myc, I could look at the expression section in GeneCards and scroll down to see the mRNA expression levels in different tissue types. You also have the option of looking at microarray data and RNA sequencing data by clicking the Show More link at the bottom of the grid. If you scroll down further, GeneCards will also have a section on protein expression levels. As you can see here, for this target, this data is not populated. If protein expression data is not available, mRNA expression data can be used instead as an indication of what cell or tissue types could be used as a positive and negative control. However, care must be taken when doing this, as there is not always a direct correlation between mRNA and protein expression profiles. In such cases, it is always best to look at other protein databases to see if protein expression information is available. The Human Protein Atlas also has protein expression data available, and other resources such as Untitled and the Cancer Cell Line Encyclopedia have mRNA expression data, which can also be checked to give additional confidence when choosing your controls. On the Human Protein Atlas website, you can view protein and mRNA expression for tissues. This can be seen on the left-hand side of the screen. There is also a section for expression data in cells, which can also be useful. This is shown on the right-hand side of the screen. As previously mentioned, androgen can be useful for looking at mRNA expression data. The same is true for the Cancer Cell Line Encyclopedia. So for my target of interest c-Myc, I found RNA expression data for different cell line types using the Human Protein Atlas. We picked K562 as a positive control and THP1 and MCF7 cells as cell lysates with a lower c-Myc expression. As you can see here on the Western blot, the protein expression observed using our antibody corresponds nicely to the mRNA expression data, which indicates that this antibody is specifically detecting c-Myc. Just to finish off, there are some cases when protein databases do not have sufficient protein or mRNA expression data due to the target being quite new. In these cases, a literature search may be necessary to choose suitable positive and negative controls. For example, here we search the literature for suitable tumour types to act as positive and negative controls for our PD-L1 antibody.